All right, welcome back, fellow scientists. So let's review. Chapter 12, we went through the first three learning objectives, right? We talked about learning objective number 1, history of DNA, Griffith and his mice and his bacteria, smooth and rough, and how he identified this mysterious transforming factor. And then we also talked about, in that same learning objective, Hershey and Chase and how they labeled the proteins and how they labeled the DNA. And they looked to see which molecule the virus injected into the bacteria, and they identified that DNA was in fact the transforming factor. Then we watched our awesome PBS video about the discovery of the structure of DNA, which happened in the 1950s. We had Watson, we had Crick, we had Rosalind Franklin, we had Maurice Wilkins, we had Linus Pauling. All of them were racing to find the same thing, the structure of DNA. There was subterfuge, there was intrigue, there was backbiting, there was drama. It was amazing. Then 3, we talked about DNA replication. We talked about why DNA needs to replicate. We talked about how DNA replicates. We had DNA helicase. We had DNA polymerase. Sounds great. Last video then, Mr. Anderson introduced us to the central dogma of biology, which is learning objective number 4, which is broken up into 2 parts, transcription and translation. So that's what we're going to talk about in the next couple videos. This video is about transcription, and then the next video is about translation. So go ahead and grab a new notebook page. I'm going to scroll down to where I want to be. And go ahead and write this down. So transcription is the process of taking the information in DNA and copying it into RNA, right? If we look at the first part of our central dogma of biology, which, if you'll notice, I wrote in Old English font, because anything written in Old English font is more important than anything that is not written in Old English font, just FYI. So it looks like DNA is turned into RNA, but that's not the case, right? I really liked Mr. Anderson's analogy when he talked about DNA being the cookbook and RNA being the 3x5 index card that you copy the recipe down onto, right? You're not changing the cookbook into the 3x5 card. You're not getting rid of the cookbook. It's still there. Right? It's safe and it's up in the cupboard, just like DNA is safe and in the nucleus. And then RNA can actually go into the kitchen and you can spill stuff on it and you can accidentally burn it, but you still have a safe copy of the information up in the cabinet. Right? RNA, it can be degraded, it can, stuff can happen to it out in the very dangerous realm of the cytoplasm of the cell. But then DNA is locked away safe in the nucleus as a good copy. Okay, so first thing that I want to do before we talk about the process of transcription is to go through and talk about the differences between DNA and RNA. So I'm going to use kind of a 2-column motif. So if you want to do that in your notebook, that'd be great. DNA and RNA. First thing is the name, obviously. So DNA, deoxyribonucleic acid. RNA, ribonucleic acid. Why are the names different? Well, if we look at the structure, we can actually see why the names are different. So this is a nucleotide of DNA. This is a nucleotide of RNA. Notice we have our sugar, we have our phosphate, we have our base. It's simplified. The base doesn't have the 2-ring structure. The phosphate doesn't have all those oxygens coming off it, but that's okay because that's not the reason why the names are different. The reason why the names are different is in the sugar, right? Deoxyribose is a sugar, and ribose is a different sugar. Remember, os means sugar, ace means enzyme. And so if we look at this number 2 carbon right here, we can see that DNA just has a hydrogen coming off of it, and that RNA has an oxygen and a hydrogen, an OH group, coming off of it. So deoxyribonucleic acid is missing 1 oxygen. That's why they're different. Now, coincidentally, this makes DNA more stable than RNA. Right? Current evolutionary thinking is that RNA was the very first information-carrying molecule because RNA can be used to make proteins. RNA can also be used to make DNA. RNA has self-splicing and self-catalyzing activity. And so that was the first one. But there was also this mysterious molecule called DNA around, and DNA and RNA could actually interact. And DNA, over the course of evolutionary history, was chosen because it's more stable, was selected for because it's more stable. So anyways, we'll get into that more in AP biology in 2 years when you take that, but just kind of tuck that away in the back of your mind for 2 years from now. So that's one difference, the name, deoxyribonucleic acid versus ribonucleic acid. DNA is obviously a double helix. We already talked about that. RNA is most often single-stranded, like 95% of the time, right? There's one type of RNA, tRNA, that's double-stranded for some portions, for some length, but we'll get into that in the next video. All right, so go ahead and write that down. That's the difference. Other difference is that there's one type of DNA. It's just a double helix. It's just kind of straightforward. Right? There's actually three types of mRNA. Or sorry, there's three types of RNA. One type of those is mRNA. That's the most famous type. So messenger RNA, tRNA, transfer RNA, which is the kind of RNA that can sometimes be double-stranded. And then rRNA, ribosomal RNA, guess where that lives? Yep, good job, in the ribosome. And we'll talk about that more in the next video. So all three of these participate in the process of translation. Right, another difference, this is key. Bases. So DNA has the bases A's and C's and T's and G's. Obviously, we already know that. RNA is a little bit different, though. RNA has A's and C's and G's and U's. Hey, Mr. Leo, how in the world does the U fit in? Well, check this out. So if RNA pairs with RNA, like it does with tRNA, then A pairs with U. Okay, easy peasy. U just replaces T. If RNA pairs with DNA, which is what we see in the process of transcription, then you gotta think a little bit, right? So if there's a T in DNA, right, T usually pairs with A. RNA has an A, and so T in DNA pairs with A in RNA, right? Perfect, good. Now, if there's an A in DNA, A usually pairs with T, but there's no T in RNA. So an A in DNA pairs with a U in RNA. Mr. Leo, you lost me. I'm sorry. Don't worry. We'll draw it out in the next slide. We'll come back to it. So just kind of be aware that that's a thing, and you need to pay attention to that. And then last but not least, last difference. Go away. There we go. Is that DNA, of course, lives in the nucleus. It's safe and it's protected, just like the cookbook up at the cabinet, safe and protected. Okay. RNA does the dangerous work. It's made in the nucleus. It has to be made in the nucleus because that's where DNA is. But then it works in the cytoplasm, out in the dangerous environment of the cytoplasm, right? And so that's where the ribosomes are. Because remember, the central dogma of biology is RNA to DNA to proteins, and proteins make you, right? And so obviously proteins are made by the ribosomes. Ribosomes are out in the cytoplasm. Ribosomes are part of the rough endoplasmic reticulum. So the RNA has to go to the ribosomes, and then the ribosomes make the protein. That's how the RNA makes the protein. That's why the RNA has to be in the cytoplasm, because that's where the ribosomes are. Perfect. All right, good. So now we have the differences between DNA and RNA. So now let's go to the process of transcription. So go ahead and write this down. We're going to use the same motif that we did with DNA replication, the before, during, and after. Before, the DNA looks the same. I even used the same sequence that we did before. Our base pairing rules still apply. All is right with the world. During is a little bit different. Okay, so during, we still have our little bubble. It's not called the replication fork anymore. It's just kind of, you know, what RNA polymerase does. We only have one enzyme, where with DNA replication we had two. So go ahead, after you have drawn our DNA with our little bubble, right? Go ahead and draw a little RNA polymerase in there. Okay, and RNA polymerase, it does it all. Okay, with DNA replication, we had helicase, which unwound, and then DNA polymerase, which made the new DNA. Right, RNA polymerase, it unwinds the DNA itself. Then it pairs RNA nucleotides with one strand of the DNA double helix. And then after the nucleotides are paired, then the copy of RNA kind of snakes away, and then the double helix just kind of like zips back up on its own. Okay, so go ahead, pause the video. What I want you to do is I want you to make an RNA strand that's complementary to this DNA strand, right? Remember, RNA doesn't have a T, it has a U. So go back and use our base pairing rules. Go ahead, do that. Okay, perfect. So hopefully you got something that looks like this. Hopefully you remembered, okay, if there's a T, it pairs with A. Perfect. If there's an A, though, it pairs with U. So this would be the mRNA molecule that RNA polymerase would make from this DNA. And then, as I said, once this is made, then that kind of snakes away, and we get to our after sequence. And so that snakes away, the DNA double helix zips back up, and then this mRNA goes to the cytoplasm. So that's the whole process of transcription, right? It's fairly straightforward. So this has been Mr. Leo. Today we talked about the process of transcription. Thank you so much for watching, and I will see you on the flip side.